What's up, world? It's Friday, May 4th, 2018. I'm here at dailymail.com. As you can see, the title is Scientists Create an Embryo from Stem Cells Without Egg or Sperm in Provocative Research. Now, this is the first of a few articles I want to go ahead and share with you today. Two different kinds of stem cells were combined and grew into an early embryo. This could create an unlimited supply of identical embryos for medical research. Embryos failing to properly implant in the womb is a leading cause of infertility. Critics argue the technique may lead to cloning, which is totally forbidden. Scientists say it is a relief the technology will not work in humans for decades. This article was posted on May 2nd, by the way. The other two articles were posted May 3rd. Scientists have made a major step toward creating life in the laboratory without using sperm or eggs. Two different kinds of stem cells were combined in a dish, and they grew into an early form of embryo. Creating embryos from stem cells would create an unlimited supply of identical embryos, which would be useful for medical research. The development is hoped to shed light on one of the biggest causes of infertility, embryos failing to implant in the womb. It will also be useful for testing the effects of new medical treatments. May lead to human clones. Researchers say that the technique carried out in mice could result in a mouse being created without using sperm or eggs within 3 years. But experts warn that if the technique could be replicated in people, it may lead to the creation of an army of human clones. It could take as long as 2 decades before the technique is likely to be perfected to create human embryos out of stem cells, the experts warned. How does the technology work? Researchers combined two different types of embryonic stem cells from mice. Once combined in a dish, they grew into the early stage of the embryo before it implants in the womb, called a blastocyst, a hollow ball of cells. When transferred into the womb, the cells initially triggered changes in the womb like those created by a normal embryo aged 3.5 days old, but failed to properly implant. Decades before technique will be used in humans. Professor Nicholas Rivron of Maastricht University, the lead researcher, told the Daily Mail that a viable mouse embryo could be created as soon as 3 years, but a human embryo would take decades. The main use for the embryos would be drug testing and research into infertility. He said, as you know, embryos are very precious and it is impossible to test drugs on them as you don't have the numbers. With blastocysts, you can open up the numbers. This will allow screening medicines in the future. He added, I do not believe in using blastocysts for human reproduction. This is ethically very questionable. This would be clones of somebody who is already alive. Human cloning is totally forbidden. Asked what the stumbling block is that stops the ball of cells implanting, Professor Rivron said, we actually don't know. This is a fascinating question. They look extremely similar to normal blastocysts and generate many cell types. However, the cells are not properly organized. They look like a disorganized embryo. Hopefully we will discover in the next 3 years. This is the first time we create structures in the lab from stem cells which have the potential to form the whole organism. The whole organism includes the baby and the extra tissues, the placenta and the yolk sac. This is the first time as far as we know. Relief. It does not work in humans. Professor Robin Lovell Badge of the Francis Crick Institute said that while the development was a breakthrough for research, it may come as a relief. It cannot yet happen in humans. He said, it may come as a relief to others that such a method of producing many genetically identical human embryo-like structures that might be capable of implantation is not feasible, even if it would be illegal to implant them into women, as is clearly the situation in the UK. Professor Rivron said it was extremely unlikely the technology could be used by a rogue state to create an army of clones, as they would not have the scientific expertise to do so. Dr. Dusko Illich, reader in stem cell science, King's College London, said this is the first time that scientists were able to shed a light on molecular mechanisms of implantation, and these findings may help us to understand more about some aspects of infertility and improve outcome of assisted reproduction. The research was published in Nature. So I just want to go to the next article here from mirror.co.uk or mirror.com. Stem cell biological breakthrough as scientists craft embryo without egg or sperm. We've just taken a step towards creating life in the laboratory. This article was posted May 3rd. Just going to read some of this, probably not the entire article. Scientists have announced a biological breakthrough by growing an embryo without either a sperm or an egg. Using two different types of stem cells, a team of researchers controversially grew them into the early stages of a human being in a petri dish. This particular research opens the door to potentially creating identical embryos that can then be harvested for yet more stem cells, keeping the cycle going indefinitely. It would allow further research on new medical treatments as well as shedding light on why some embryos fail to develop in the womb. But it could also draw complaints from some who see it as playing God, which in fact is what it's doing. The experiment was carried out by scientists from the Merlin Institute at Maastricht University and the Hubrecht Institute in the Netherlands. Our research helps to understand the perfect path an early embryo must take for a healthy development, said Dr. Nicholas Rivron, who led the scientific team. Professor Clemens van Blitterswyk, department chair at the Merlin Institute of Maastricht University, added, "This research opens up the path to a new biomedical discipline. We can create large numbers of model embryos and build up new knowledge by systematically testing new medical techniques and potential medicines. It also dramatically reduces the need for animal experimentation. There's no danger of this science spreading into the realm of human cloning. The practice was made illegal in Europe by a parliamentary resolution in 1997. If it was illegal in 1997, then they must have been doing cloning back then, right?" I'm going to go ahead and just briefly show this next article here, which is from express.co.uk or express.com. Cloning horror. Human clone fears as Euroscientists create life from nothing. And again, this article was posted May 3rd. Scientists have created a living embryo in a laboratory without using either egg or sperm in groundbreaking but hugely controversial experiments. There is a short video here. I actually have the full video, and I will be sharing that with you in just a few minutes, and it's about 4 minutes long. I'm not going to read any more of this article. It's pretty much the same as what we just read, but I'll leave a link for this article as well. I just want to go ahead and mention a couple of things before I show that video that I mentioned. Some of you may have seen or heard of the movie The Boys from Brazil. It was released in 1978. Really an old school movie, you know, there's no special effects, nothing like that, and it's pretty much all about the storyline. I have seen the movie, and I suggest, if you're interested in learning about cloning or researching cloning, I would suggest taking a look at the movie. If anyone caught it, some of the research that was being done is by the Merlin Institute at Maastricht University. Here at ancientorigins.net. It's a little article here on the legendary origins of Merlin, the magician. And I just highlighted a couple of things here that I wanted to include in this video. The powerful wizard is depicted with many magical powers, including the power of shape-shifting. And we all know that shape-shifting can be linked to reptilians. Merlin was paradoxical, as he was both the son of the devil and the servant of God. Therefore, Merlin was part a creation of God and part a creation of the devil. And it's actually said that an incubus was who fathered Merlin. Just an interesting thing to throw in there. So I'm going to go ahead and share a video with you. I believe it was here on Mirror. No. This video here at Express, I'm going to share with you. And I'll also share a clip from the boys from Brazil as well. So that will conclude this video. Everyone leave your comments and opinions. Take care. God bless.
After years of research, the laboratory of Nicolas Rivron has found out how to induce stem cells to organize themselves and develop into an early embryo-like structure in a dish, all in the lab. This scientific breakthrough has truly opened up the black box of early pregnancy.
Surprisingly, we know very little about the first days of pregnancy. At that time, the embryo is extremely small. It's about the width of a hair, and it's hidden in the womb. It is floating inside the uterus. It contains less than 100 cells, and the cells from the outside are the stem cells that are going to form the placenta. Inside, there is a very small cluster of about 10 embryonic stem cells that are going to form the whole embryo. We can easily harvest, culture and multiply stem cells in the lab. We can do this for very early placental stem cells, very early embryonic stem cells, and they multiply while remaining locked in time. But in a natural embryo, those stem cells, they are together in 3 dimensions. And those stem cells, they are chatting, they are talking to each other, a language that we barely understand. What we did is to place them back together in a specific cocktail, and they started to converse. They started to spontaneously organize. So it's only mouse stem cells sprinkled with a handful of molecules that are self-organizing. This is like self-organizing life. Those embryo models are remarkably similar to mouse embryos, to the point that when transferred back into the uterus of a mouse, they implant and initiate a pregnancy. However, we clearly do not form a full mouse. These are just very useful models to research in the lab. So now we can form those model embryos in very large numbers, and we can study them in great details.
And through studying these early embryo models in the lab, we can achieve a great deal. We can finally precisely understand how adult diseases start within the embryo, thus enabling us to prevent and reverse their appearance. We will also be able to discover new medicines without animal experiments. For instance, medicines to help couples who experience difficulties when it comes to having a child. All of this begins with the same thing, understanding the communication between stem cells.
What we observed is that the embryonic cells are the chatty ones here. They are the ones that are instructing the placental stem cells to multiply, to organize, and also to implant into the uterus. So this stem cell conversation that we seized opened very new perspective to research in the lab the problems of infertility or pregnancy failure. For example, 2/3 of the in vitro fertilization treatments are currently failing, and they are failing at the time of implantation in the uterus. And we just don't know why. Using those model embryos, we can test and develop new molecules for the embryo to take a perfect path for a successful and a healthy pregnancy. These new molecules that we could find might become the medicines of the future.
Twins.
Then he was nothing more than a sadist, really.
A sadist with an MD and a PhD?
Well, some people would say that's a perfect definition of a scientist. What exactly do you mean when you say the boys you saw were more than twins?
Not only did they look alike, but they were also very alike in personality.
That is unusual. Studies show that twins who are separated at birth develop totally different personalities.
But these twins, or perhaps I should say triplets, because I believe that my associates saw another, were like the same people, but brought up with different languages.
It's impossible, of course.
Excuse me, doctor, but what is impossible? What is impossible, doctor?
Mononuclear reproduction.
Oh, doctor.
Cloning.